So as you all know, that when we sequence a microbial community data, we may have hundreds of samples, and we may have thousands and thousands of OTUs or ASVs or whatever, right? The point is that when you get a data set, you have hundreds of these members that may be correlated in some way, right? So the point of doing co-occurrence network analysis, at least the co-occurrence part, is that we can get understanding beyond sample level, beyond the alpha diversity, beta diversity level. We can understand whether some members, they like to associate with each other, they like to share same niche, all right, or they don't like to, they are more competitors, they don't like to be associating with the same niche, you know. And we can find how why some of these members, they co-occur together. What are the environmental parameters that stimulate their co-occurrence or their co-abundance, right? And perhaps some of the network level scores, some of the statistical scores that can be used towards identifying these hub taxa or keystone taxa that are highly correlated they are more influential in the community. Okay, now I would like to point out here that here we are talking about network analysis, co-occurrence analysis, but network analysis can be based on correlations. So always correlation is not causation. So correlation-based analysis must be validated empirically. So we need to keep that in mind. So the notion of keystone taxa, as previously was mentioned, what is keystone? So as you all see this arch on your left, this critical stone in the middle is called a keystone, because if you take this stone out, the idea is that the whole arc is going to fall apart. The notion of keystone taxa, it was originally proposed by Dr. Robert Payne when in 1960s. In 1966, he was actually working in the shoreline in Washington, in MICROBE. And what he did, he selectively removed sea stars, disaster. He removed these sea stars from the shoreline. And what he found was absolutely incredible, that just by removing a single species from this shoreline, the diversity declined almost to its half. So just by removing one species, the resident diversity almost declined to its half. So, and then after that, he coined the term keystone taxa. There are many other examples of keystone species in the animal world. For example, African elephant, or a hummingbird in a desert, or perhaps beavers. These are all ecosystem engineers or highly influential players in their own community, because they can alter their ecosystems. So they hold more importance in the microbial world, or in the animal world, actually. Right. So the notion of keystone taxa originally came from animal world. I have done some. I have used statistical bed correlation analysis, network analysis, to identify keystone taxa. And one of the studies that I would like to point out here was something that was done in Switzerland while I was working in Agroscope in Zurich. What we did, we looked at the root microbiome networks, and we looked at root microbiome network under 3 different management practices. One was conventional, where they do not apply any fertilizers or synthetic pesticides. Where they do apply chemical fertilizers and synthetic pesticide. Another was no-till, where they do not till the soil, and there was organic, that do not apply. They don't apply any synthetic fertilizers or pesticides. What we found was very interesting, that under conventional farming, it was a very simple network, as you can see on your left. Same with no-till farming, it was also a kind of a similar kind of a simple network, not so much going on here. There are a couple of these diamond-shaped keystone taxa. Actually, I was going to talk about it. So, and then on your right, you see this organic farming network, where it's a much more complex network. Although the number of nodes is kind of similar to conventional and no-till, but if you look at the number of edges or the number of associations, it's 2 times more than conventional and no-till. So what we found was interesting, that this network is rich because it had a lot of these diamond-shaped nodes, which are the keystone taxa, And all of these, in majority of these keystone taxa belong to mycorrhizae. So belong to the order Glomeralis, Paraglomeralis, Diversisporalis, and so on. So all these mycorrhizal keystone taxa actually made these network very rich. We published this article a few years back where we compiled a list of keystone taxa that have been identified across different microbiomes. And we also proposed this definition of microbial keystone taxa. We said that, look, microbial keystone taxa are highly connected taxa that individually or in a group, like in a guild, can exert considerable influence on microbiome structure and functioning, irrespective of their abundance across space and time. So their actual influence doesn't have to be proportional to their abundance. They can be highly abundant, and they can be less abundant. But the point is that they will exert more influence on their community across space or time. And we found that in the human microbiome, they are already empirically validated keystone taxa. For example, Porphyromonas gingivalis that are linked to gingivitis is actually a keystone taxa in the oral microbiome. For example, Helicobacter pylori, Bacteroides vagialis, and Ruminococcus bromis. These are all empirically validated keystone taxa in the gut microbiome. Similarly, for the plant microbiome, when we published this study, there were not so many empirically validated keystone taxa, but there were a lot of these statistically identified keystone taxa that have been statistically identified through network analysis. And we found that a lot of these, in many cases, a lot of these keystone taxa belong to the order Rhizobialis and Burkholderiolis. So there were many cases there are keystone taxa belong to these specific orders. So why we need to, I think, it was mentioned, why do we need to identify keystone taxa? Why do we even care? So imagine going back to this human society example. We are consistently interacting with each other, and we live in a community, right? If we want to say we live in a community and we want to have this rule that from 10 o'clock at night to 6 o'clock in the morning, we want this to be a quiet hour, so there should not be any loud noise or something, right? Are we supposed to go to individual committee members and tell individuals, say, if this committee has thousands of members, we're not going to go to individual committee member and pass that information, right? What we can do, perhaps, we can find the community leader instead and tell him or her and say, look, this needs to be the rule, right? So basically the same notion applies, that if we can identify this influential pair in the microbial community, our job becomes a little bit easier, because in that case we can identify these members, and through them would be able to manipulate the community. We'll be able to, because the community leaders, that they can disseminate the information. So that was the idea of that is why we need to identify these keystone taxa or these influential member in the community. Something that we need to remember that it was previously mentioned, whether these members are functionally redundant, we don't know. There may be, whether these are male members, female members. Just to give you an example, in the leader of the community, what kind of member, we don't know. Whether they are replaceable, whether they are redundant to the community, whether somebody else can take their place, and so on. We don't know that. But that needs to be investigated, because the potential is there for microbiome manipulation and microbiome management. If you can identify these influential players, our task becomes a little bit easier, because then we have to just manipulate these members, and we could be able to use them as conduit, and we'll be able to manipulate the microbial community. So in my lab, in my current lab, what we are trying to do, we are trying to identify keystone taxa in stress microbiome. Because in stress microbiome, the goal is to identify, are there members that can hold the community together when you are in a stressed environment, right? And one of the stressors that we are working on is salinity stress. Soil salinization is a huge problem in the world, around the world, because nearly 33% of all agricultural land, and 20% of all cultivated land across the world are affected by soil salinity. And it's supposed to increase up to 30%. The number of cultivated land actually that going to experience the soil salinity stress by 2050 is going to increase by 30%. And already over 1 billion hectares in 100 countries is affected by salinity stress, and the number of the crop loss is billions of dollars we lost to soil salinity. So it's a major stress because it affects plant growth, crop growth. It's a major osmotic stress because it affects the nutrient deficiency. It causes nutrient deficiency. It affects the nutrient acquiring of a crop, and it's a major oxidative stress as well. So because of that, we are trying to identify keystone taxa as a way to manipulate the microbiome under salinity. So what we did, we conducted two different studies. We did an exploratory study where we looked at 4 different field trials. So in the northern Great Plains, we looked at in North Dakota, we looked at 4 different field trials. We published this study last year, but basically this was an exploratory part of this work where we looked at the saline soil compared to non-saline soil, and we identified keystone taxa statistically using network analysis. And then after that, we also did a manipulative study where we had 3 different levels of salinity. A low salinity where is 3 decicements, so basically it's a low salinity, and medium salinity where it was around 6 decicements, and then we had high salinity where it was around 10 decicements. And we also had control plots with plants and double negative plots without any plants. So after that, we
just to remind you, have 5 minutes just to not to be unpolite, just
oh, yeah,
that you have a feeling.
Okay.
Thank you.
Right. Yeah. So from these samples, we did amplicon sequencing as well as metabolomics and metagenomics to identify how not only to identify keystone taxa, but to see how the communities are and the root exudates profile changes in different salinity stress, and are there important members, are there certain influential member in the community. And we found that indeed the community structure was quite different between saline and non-saline environment, both in rhizosphere microbiome as well as in root microbiome. And that changes across different time points, because we also had 5 different time points. We found that indeed the community changed based on different salinity levels. And when we looked at the root exudate profile, we also found that indeed the root exudate profile was quite different between the saline and non-saline samples, non-saline environment. When we are crops under wheat plants under stress, their root exudate profile was quite different when we looked at using metabolomics. So after that comes to the mechanistic validation part, where we looked at these networks under low salinity and high salinity. As you can see, under low salinity, the network is much more connected, a little bit more complex compared to high salinity, but nonetheless, we identify some keystone taxa in both of these communities. And then what we did, we established a culture collection. We identified many of these top 30 of these members, and then we also identified some peripheral taxa from these networks. And then after that, we so basically the idea was that we established this SYNCOM using across 3 different levels of salinity, and we used all these different agar, LB, TSA, and soil extract agar to establish a culture collection. We had over 340 strains, and we extracted the DNA, and when we sequenced these members, and then, well, actually it was 323, and we identify all these members from this community. We established this culture collection. and then we selected these microbes. We matched from our culture collection. We from our synthetic community, we identified which of these members actually was the keystone taxa in the network, right? We found that these were the keystone taxa. A lot, there are 3 different bacillus members, and there are some other members. These were the keystone taxa in the salinity networks, as well as there are some peripheral taxa that we also identify, and we looked at the 100% identity and highest B score and lowest E value. So what after that we did was that we looked at how quickly they grow, because if we were to grow them together to do an experiment, we looked at how quickly they grow and their cell culture. And we used flow cytometry to count their cells, and then we just completed a dropout experiment where we are doing what we are doing in presence or absence of this keystone taxa, how the network connectivity and how these members they sort of change. And this experiment, we just completed this experiment, so it's still underway. So I just wanted to say that we just completed this experiment. So we are still waiting on the sequencing results. So I can't, we don't have the results yet, but we are, we conducted this experiment with and without keystone taxa that we identified from this first using statistical correlation, but then from our synthetic community. And we are currently waiting on these results. But basically, we would like to, what we want to see, that whether these members can hold the community together, make the communities more stable or more connected under high stress. So that is the whole idea of this work. So again, as I mentioned, that we are still waiting on these results, but I truly see the opportunity that we can identify these. If we can identify these influential players in the community, we would be able to manipulate the microbiome, and we can see if these members actually hold the community, make the community more stable when they are under stress. So with that, I would like to thank my lab members for their work, and all these funding bodies, and I would be happy to answer any questions. Thank you.
Yeah, hi, thank you for the talk. Just for my understanding, so I understand that you grow the 14 strains together under laboratory conditions. Are you planning then to insert them or to add them to the rhizosphere and then see what their effects were, or what was exactly the procedure or the plan?
These 14 strains that we just discussed, yeah. So the first was to see that ultimately what we want to see, the connectivity, how the connectivity changes over time. And then we here in this study, in this dropout study, was just presence and absence. But ultimate goal was to introduce the keystone taxa at different levels of salinity. First in a microcosm, maybe in a tube, and to see if the connectivity remains the same when the keystone taxa is present in the community. Like, when under high stress, the keystone taxa, when they're present in the community, if they can make the community, keep the community connected or more stable, even under high stress. Now, the final goal, of course, to see if we can find some beneficial members or some of these keystone taxa that can also provide salinity tolerance to their host. Then we can perhaps apply them to in the greenhouse to a pot to see if they can also help the plant to be more salinity tolerant, to tolerate the in a way to kind of alleviate salinity stress on the crop, and see if there is a way that we can use these members to sort of help the host plant. But that's, I guess, the future direction, I guess.
Yeah, maybe I simply start. You but also others identified often bacilli, pennybacilli, whatever, as keystone species. Couldn't this be also related just to the lifestyle, that they form spores and they are quite often present in high abundance, independent if they are active or not? So this is a little bit what I'm a little bit concerned about, this DNA-based analysis, that we simply don't know what it means, and mainly for this bacteria which can forms like spores. It is always very difficult to predict if they are really active or if they are just there providing at least the potential, which is also, of course, very important. But if they are really active at a certain point, I think this is difficult to assess from the DNA-based data.
Yeah, that's a great point. So in our culture collection, we had some of the, of course, some of the Bacillus member, but we also had some other members as well. When it comes to Bacillus member, absolutely that concern that you expressed, it's definitely a legitimate concern. That could be a possible cause because of the spore formation. Whether that makes them that will make them make us sort of identify them as keystone taxa, we don't know that. But whether they are because that's why we're waiting on these results. If we can see that their presence can keep the synthetic community connected under high stress, then the idea would be to look at what sort of metabolites they produce, perhaps to sort of see if it's just a kind of a like a correlation or an artifact, or is actually a real thing. So we do have other members. We do have members other than Bacillus. Yeah, I guess it's definitely a concern. But because we have some other members as well, we'll keep this in mind to see whether it's because of the spore formation or actually the true importance, where they are secreting some metabolites or secreting some kind of chemical compounds in order to selectively modulate the community and making the community more resilient under high stress.
Thank you. Mine is just a quick question. Thank you very much for the presentation. I was interested in the dropout experiment, and I was wondering which plant are you using for this experiment? I think I missed that. And what is your control in the experiment? How do you control the experiment?
Right. So this is without any plants. So we haven't gone to the plant part yet. So this is still in tubes. So this is still in sort of in microcosm. So we haven't gone to the in presence of a host plant yet, because first we want to do some mechanistic validation in the lab and then take it to the greenhouse maybe. So that's the number one. And number two was the control was, you see, so these are the peripheral taxa, and these are the, so our control was just the peripheral taxa. So one where we only had the peripheral taxa, another was just the medium itself. So those are the two different controls that we had. Then we had these different strains added to this peripheral taxa to see how the connectivity changes in presence of these different keystones. So the control here was just a medium, was just the broth, and here we only have these peripheral taxa, and then we also had different keystone taxa, inoculated in different tubes, and then we monitored over across 5 different time points over 120 hours, and then we had all these samples that we sent for sequencing. So basically our control, this is our double negative control. And then we had this another control as the positive control as the peripheral taxa.
So I would like just to ask you perhaps a little bit more philosophical question based on your experiences. So is the taxonomical information really enough to define the keystone taxa? I mean, your example here now is based also on ASVs and bacteria only. Should we go metagenomic? Should we go functional? How far do you think really we should go? Or do you really think that taxonomy itself is enough, even though, what is a bacillus or what is even Bacillus subtilis? So I would just like to hear your opinion on even a little bit perhaps future oriented if, technologies and prices become even more reasonable. Thank
No,
you.
that's a great question, actually. I don't think only taxonomy is enough. When it comes to keystones and understanding the true importance, I don't think only taxonomy is enough for this. Okay, I do think eventually in future we need to go towards microbial chemistry to identify what sort of compounds they secrete. How do they modulate? If we can identify these true leaders of the community. Now, again, these leaders doesn't have to be the leader always. They can be the leader of that particular space or that particular time. But the point was that how do they exert the control? How do they selectively modulate the community? For example, in the network, if you look at a network, you will see in a network that keystones are connected to their subsidiary taxa. Through this subsidiary taxa, they can influence the peripheral taxa, right? So basically, the point is they can selectively modulate these subsidiary taxa. So how do they do that? In order to identify that, in order to understand that, I think we do need to do metabolomics as well as metagenomics to see what sort of functional level information we can glean from this. That information we can glean from this, and then eventually, if we can identify some metabolites or some chemical compounds that are consistently present when that particular keystone is present. So I think that's where the field is. That's what I see happening in future.
So Dr. Salma Gomez Vieira is currently postdoc at Leibniz Institute, DSMZ, the German collection of microorganisms and cell cultures in Germany. Her work is based in a huge dataset from high-throughput sequencing data of soil samples using co-occurrence network to infer microbe-microbe interaction. So today she will talk about the identity, activity, and niche preferences of microbial keystone taxa in complex soil ecosystems. Salma?
Okay, can everybody hear me? I guess so. Can everybody see my presentation? Great. Thanks a lot, Pamela, and for inviting me to be here to present my work. One second. So my work is about pinpointing microbial keystones in soil ecosystem and also assessing their activity and niche preferences. And this is done in the scope of the biodiversity exploratories, which many of you are already familiar with, but I thought I would briefly introduce, and also the project I'm part of. Can I go? Yeah. Okay. So the biodiversity exploratories is an research infrastructure in Germany for research on biodiversity, and it is mainly focused on the interplay between land use, biodiversity, and ecosystem processes. So how different land use practices and intensities affect biodiversity in the ecosystem processes. How the different biodiversity aspects are interconnected and interact between each other, and how they affect then the ecosystem processes. So this has been set up in 3 different regions in Germany. So we have in total 300 plots. So in the north, in the Biosphere Reserve Schorfheide-Chorin, we have access to 50 forest plots and 50 grassland plots. In the middle of Germany, in the National Park Hainischen surroundings, we have the same setup of 50 forest and 50 grassland plots. And again, in the south, in the Biosphere Reserve Schwäbisch Alb, the same. So at first, so we are involved in the biodiversity exploratories in a project which is a cooperation between us here at DSMZ, partners in the Helmholtz Center for Environmental Research and also the Technical University of Munich. And this is one of the central projects in the biodiversity exploratories, and we do not only research but also provide some services to the biodiversity exploratories communities. So we provide long-term data on diversity of soil bacteria, fungi, and archaea, and also we try to identify the interaction between these different microbial groups. We provide data on functional properties of complete soil microbiomes through metagenomics, and also, which is more relevance for the topic today, we are trying to pinpoint keystone species, their activity, and also their functional properties. Moreover, we also provide, we are a central bank for DNA and RNA extracts from soil samples from coordinated soil sampling campaigns, and we store also some small amounts of soil samples for molecular work. We have been working in the exploratories for a long time now, and so since we have samples since 2011, and there are huge coordinated sampling campaigns every 3 years that involve a lot of people, and where all the 300 plots are sampled more or less simultaneously in spring. So we have that time point 2011, 14, 17, 2021, and 23. But the samples from last year's campaign are not yet processed completely, at least. So the work I'm presenting today here is regarding 4 different time points. As I said, we have these 3 different regions and 2 biomes, so grasslands and forests with 50 plots per set, which gives us 1,200 bacterial and fungal amplicons. So we use the V3 region of the 16S rRNA as a marker for bacteria, and the ATS2 for fungi. And this is a huge diversity and a huge data set. You see here that for bacteria, for example, we have over 450,000 sequence variants, and for fungal a little more than 13,000. So first and foremost, we see that the communities in these soils are heavily influenced by edaphic properties. Here you see a picture of a principal component analysis on just the soil properties, so physical chemical parameters. And you see, for example, for one of the regions, they cluster by soil types. In one hand, histosols and glazols, and that group there in red, and albiluvisols, luvisols, and chemisols in the other group in green. And we see that, for example, bacteria mimic this pattern. So bacterial community structure, you see also grouping in the same way. And it's not shown here, but we know that pH is the major driver of these communities, as we see also in studies worldwide. So this is nothing surprising. We also had a look on the effect of land use on these communities, and in contrast to what our colleagues see for macroorganisms, the microbial communities in these soils do not seem to be affected by the different land use. So this is both true for bacteria and fungi. In these plots, we see them. For example, the land use index for forests in the x-axis and plotted against bacterial regions, and we see that there is virtually no relationship here. However, one has to say that there is no intense land management like one would see in agricultural fields in the exploratory, so this may be why we don't see any effects of land use in the below ground communities. So we also see, very importantly, that these communities are fairly stable over time. So this is both so more so bacteria than fungi, but still communities are stable over time. Here you see plotted richness for bacteria for the Schwäbisch Alb forests and grasslands, and we see that it's more or less stable over time. There is somehow an increase there on the point of 2021, but this may be a little bit of a technical artifact, because we have a much bigger sequencing depth here for these samples. But if we have a look at the beta diversity, also community structure, we see that these communities are fairly stable over time. Yeah, so and this is a very important point here. So then we come to the concept of keystones, because we want to identify who are the key players on maintaining the stability of these communities. So we know that there are a little bit of different definitions for keystones. So more simple definitions that involve a strong influence on the community stability and structure, but also definitions that involve a disproportionate effect relative to abundances or biomass. But the data that we have and that many people have do not contain absolute abundance data or biomass. So we are operating here on the more simplistic definitions. And as has been pointed out before, how to approach pinpointing keystones with this amplicon data, and one tool that we can make use of are network analysis, so either co-occurrence or correlation networks, and this is what we used in this study. So here we constructed networks for all these different environments, so split across regions of the exploratories, the different biomes and time points. And after cropping our data sets a little bit, we constructed correlation networks with an algorithm that takes into consideration compositionality of the data and also sparse data. And so we came up with a lot of different networks, and we see that some are much larger. For example, the grassland network seems to be a little bit larger than forests, and in some areas, for example, shore-fire forests, we see that the networks are quite small, but this is also related to the richness in these areas, that so it's already lower, the richness in these areas when we compare to other forest environments, for example. So after network generation, how do we go about selecting and pinpointing keystones? So here we identify keystones running a model through these networks. So we run a model in the normal network, and then we disturb the network by removing one node at a time, and assessing the impact that the removal of this node or this sequence variant had in the structure of the network. And then we can rank the nodes by importance. So we did this, and since we know that the networks are rather stable throughout the years, we then selected keystones for each biome independently of the year. And so we selected the top 25% of these rankings. So the sequence variants that were present in the top 25% of the rankings in at least 3 out of the 4 time points, because we wanted to be quite conservative here. We rather have less, a smaller keystone list, but that we are fairly sure that they are the important ones, than have a larger set. So we also, very importantly, tried to match this data to activity. So for bacteria, for the set of 2021, we have both RNA and DNA-based amplicon data sets, which allows us to calculate 16S RNA to rDNA ratios. And importantly here, these were corrected for stochastic variation in sequencing by a method that my colleague Johannes Sikorski has developed. So in the end, we have a list of active taxa present in these samples, and activity should be a prerequisite for keystone species, and this was already mentioned in the first definition that Robert Payne gave in 1969. So we then cross-match this active list of active taxa against other keystone taxa lists, and we see that keystones among keystones, there is a higher percentage of active taxa than among non-keystones. So you see on the graphs on the left, there are increments. So we go through the ranking of keystones in increments of 5%, and then on the y-axis you see the percentage match to activity, and you see that at the top of our rankings there is a much higher match to active sequence variants than when we go to the bottom of these rankings. And on the right you see the aggregated data on this, and this is true for both forest and grassland bacteria. Sadly, we don't have the same data for fungi, so we can only say this for bacteria, but it is true that these keystones that we selected seem to be more active, which validates, we think, we believe, our method. So we have now a list of microbial keystones, and we see that they are very diverse. They span nearly the whole tree for bacteria, at least. And the same is true a little bit for fungi. Here you have a depiction of the grassland bacterial keystones, which is the largest set of 217 keystones. But so we wanted to check first if there is some kind of phylogenetic signal for keystoneness, let's put it like this, so for being a keystone. And so for this we measured 2 different metrics that allows us to pinpoint a phylogenetic signal at either deep branches or terminal branches of the tree. And it seems that for bacterial keystones, they tend to cluster at terminal branches, so there is some phylogenetic signal for it being a keystone. Moreover, we try to understand. We don't have, sadly, functional data, so we try to at least understand to which niches these bacteria or these fungi are adapted to. Do all the keystones have the same niche preferences, or do they occupy different ones? So for this, we model their abundance against the range of environmental parameters that we have that are listed here. And this here on the graphs on the right show you examples for pH. So we have on the top one sequence variant that, for example, is more adapted to more acidic pHs and it has a broader pH range. And in the bottom we have a sequence variant that is more that likes neutral pH around neutral values, and it has a narrower pH. So we can do this for, of course, we did this for all the keystones, and we found very different patterns. But we also see sometimes that closely related keystones seem to share some kind of habitat preferences. So we need to dig a little bit deeper here, and of course there is a lot of information to be seen here. But yeah, I just wanted to give you here a first look. So, and
Selma,
now, yeah,
Selma, just 5 minutes that you have a feeling.
At the
Thank
end.
you. Oh, okay, thank you.
So just some final considerations. So I could show you, I think that microbial communities in the source of the biodiversity exploratories at least are rather stable over time, and therefore so are their keystones, and there we can pinpoint keystones for these particular biomes and regions. Soil properties are the main drivers of these communities, and the keystones themselves are very specific to the region. So there's not many overlaps between regions and biomes. So they are very specific to their sites. Keystones are very diverse, both phylogenetically and also in relation to their habitat preferences, but there seem to be some phylogenetic signals there. And but of course, one big open question remains in regards to their function, of course, and we are always striving to go further here, and we are involved in doing bacterial cultivation to hopefully try to isolate these keystones and understand their functions, their traits, and also in this area we are trying to go forward with long-read metagenomics, to hopefully in the end answer these big questions about their function in these communities. So now I just need to to thank a whole bunch of people. I'm not going to name them one by one, but of course everybody involved in the biodiversity exploratories and the DFG for funding the biodiversity exploratories, all the members from the team from Core 8 project microorganisms, and also the members from the Core 9 project soil, which provide us with a lot of soil data. And of course, thank you, and I'm open to any questions that you may have.
Yeah, I have a questions like regarding the differences between grassland and forest soil, because you showed like the forest soil like to have a much sparser network in
just
grassland.
Just in one of the sites. This is true, and this is because these are very acidic soils. So we believe this is the reason that they already have a lower richness when compared to other the other forests that we have.
Yeah, like my question was more like, how do you account, for example, when doing this kind of comparative analysis for differences, like in not only soil biogeochemical properties, for example, like differences in humidity, like more overall retention of water that could account for metabolites traveling longer distances, or like other properties like more on the bio side, like for example where you have a more deeply connected by, like you have more hyphae like mycelium, and then these can function as highway, putting like more specially separated taxa in.
So that is a great question. Of course, the environmental filtering is a big issue here, and that's why we have constructed networks separated by regions. So in soils that are similar in between themselves, because there is no sense into integrating everything in one network that is very different, like you have said. And of course, these are everything that you mentioned are drivers of these communities, and they are different, of course, between sites. But we don't plan to integrate this into one network. So we always keep this separated for the sites, specifically for these reasons.
But basically, what I'm going to is more like, what if you assume, for example, that the underlying complexity of the network of an ecosystem has to be somehow at a certain, like, let's say, threshold of complexity for the ecosystem to function, like to be viable. And what we can conclude from that is that when you have two very different networks, like in this case, it means that one was just not really captured by the sequencing or the analysis or everything. So that there's like some other confounding variable that is like precluding that.
You mean that why these networks are smaller?
No, I mean like that arguably, like to have a functioning communities, like you expect like the ecosystem, like the players in the microbiome to reach a certain level of complexity that would make them function. So we can assume that networks somehow should be in a similar range of complexity, let's say, which means that there's like some other confounding variable like that we are not accounting for.
This could be true, but we already know that the richness of these communities are very different. and most likely because of the differences in pHs, which is the major driver of these communities. So and our sequencing depth is fairly large, so around 1 to 2 million reads per sample. So we have, and also sequence coverage estimates are quite good. So we are fairly certain that we are covering nearly all members of these communities. So this the reason for the communities in this particular area being so small, I mean, it is like this. The communities are simpler, therefore we have simpler networks.
Let's try to make the next two questions because we are running out of time, but Michael and then Cecilia.
Yeah, thanks for this really very nice presentation about the exploratories. I was wondering a little bit about temporal dynamics. We know from single microbiota, they change in their abundance over a season in grasslands very much. You have some in spring, you have others when the grasses are flowering. So what do you think? I don't know, you have the data, but what is your feeling? And I think this is for the discussion also very important. What about the temporal dynamics of these networks? Do you think they are more stable? Based on the definition, they should be. But what do you feel about that?
I believe that they should be stable, but there is probably different states of the networks throughout the seasons. We sadly don't have other data than the springtime, but of course other players will become more important in other times of year. That's what I believe. So here what we can say, or we can show our keystones for spring, let's say like this. We cannot really say that they are always the same keystones. I don't believe that this is true, because of course in different times of year you different functions are needed or become more important.
Cecilia?
Yes. Can you hear me, Salma?
Mm-hmm.
Good. My question is a bit related to what Michael was saying. And also, I like this idea that you did in silico of perturbing the systems to challenge it to see what happens. I was wondering if it's in the plans, or maybe other groups in the exploratories are doing microcosmos experiments to do that in vivo, to alter the system by changing the main parameters on different soil samples and see what happens. At least as an initial point, in the long term is, like others have said, it's good to go to the functional analysis, but meanwhile, to challenge the system in microcosms and see which taxonomic responses you get. Is there any plans for
that? Short answer, no, not at the moment. But of course, this is an interesting question. But of course, we still don't know, or need to first come up with the notion of what is the normal microbiome here, so that we can understand what is a disturbed microbiome, and how how this changes the keystones and the players and the relationship between them. So I think we are still at the first step into understanding the temporal dynamics of these communities, but of course this is an interesting idea to follow up.
The team is a PhD student at Professor Ines Thiel's group at the University of Galway in Ireland. And today he will present this in silico modeling bridges the gap between correlation-based and experimental microbiome studies.
Thank you so much. First of all, thanks for the organizers for providing me with this nice opportunity. So today, I want to tell you a bit about the computational modeling approaches we are using to make predictions on functional phenotypes of microbiomes, and hopefully this more functional metabolic lens will provide a useful addition to the discussion later this morning. So, as I'm sure you're all aware of, microbiomes in our gut are vital modulators of complex diseases. They have been found to play roles in Alzheimer's disease, Parkinson's disease, and inflammatory bowel diseases, among others. However, the exact role of gut microbiota in health and disease is still unknown, and this is partly due to the fact that omics-based correlation methods are often unable to provide mechanistic insights. So we need more cultivation methods. However, this is not always feasible due to some bacteria being unculturable. So an alternative and complementary approach for this would be mechanistic in silico modeling of metabolic phenotypes of microbiome and host crosstalk. So this metabolic modeling approach is based on so-called genome scale metabolic reconstructions, which are knowledge bases that integrate biochemical data with functional genomics and physiological knowledge. And importantly, these reconstructions can be converted into computational models, which can be used to make predictions on metabolic phenotypes. And in the past few years, there have been resources created of metabolic reconstructions of gut microbes. And recently, last year, our lab introduced Agora 2, which contains over 7,000 strains across 25 phyla. And these resources enable the interrogations and mechanistic predictions of more and more microbes, but they also enable the generation of microbiome community models, where several reconstructions are joined together. And in 2019, our lab introduced a workflow for creating personalized microbiome community models. So in this workflow, 16S or shotgun sequencing microbiome data is used to select metabolic reconstructions of microbes, which can then be combined in a shared microbiota lumen, where metabolites can be freely exchanged between microbes. And a dietary component is then added, where a personalized or a predefined diet can be added based on experimental data. And of course, a fecal compartment is added for waste removal. So relative microbial abundances are further incorporated by a so-called community biomass reaction, which kind of ensures that the emergent metabolic phenotypes that can be predicted from these microbiome models, that they are proportional for each microbial species or genus or whatever the taxonomic resolution is that you want. So microbiome modeling has been...
Tim, sorry, just 1 minute to remind you. Thank you.
Yes, oh, of course, 1 minute. So I will go quickly. So this method has been applied for several diseases. However, the current, when only taking the microbiome into account, you don't account for the host. So currently, we are working on adding the microbiome to a large-scale metabolic reconstruction of human metabolism, and we're demonstrating this at the moment on Alzheimer's disease. We have 2 papers being in preparation. And now, so for some take-home messages, we found that metabolic modeling approaches, they integrate experimental and molecular omics data, and they enable mechanistic hypotheses on microbial crosstalk, which is with its environment, which is of course due to the fact that these models are deterministic. And this approach has already been in use for 5 years now. Importantly, these in silico approaches are complementary to cultivation-based approaches, as the genome scale metabolic reconstructions, they are informed by experimental data, and the predictions of metabolic phenotypes are validated again by experimental data. So I hope this was a useful window into useful look into the world of metabolic modeling, and I hope that thinking about these about keystone taxa and core microbiota from a more functional metabolic perspective would be useful. So thank you all for listening. One final thing, we recently got a new ERC grant. So we are hiring. So if anyone is interested in working at us, please contact us. So thank you all for listening.
So Dr. Diana Andrade Linares is a postdoc here at Department of Comparative Microbiome Analysis at Helmholtz Center Munich in Germany. And today she will present the correlation network and core microbiome, a case study in pre-alpine grasslands. Now, yes.
Yes, okay, good. So, good morning, everybody. Thank you, first of all, to the organizers for this nice workshop and the possibility to share some experiences and knowledge that we have already with our study. In this case, today I will present the SUS Alps project from a technical point of view, with the idea to implement this data set of networks and core microbiome analysis on other biodiversity data set in grasslands. We are investigating the soil microbiome of these 3 pre-alpine grasslands, which are located along an elevation gradient under different climate change and agricultural management effects. We want to find out which are the keystone taxa in these pre-alpine grasslands, and also if they are part of the core microbiome. And if we have this core microbiome, how similar or different is this core in as comparing with other temperate grasslands in Germany. For this analysis, we will first need to analyze the microbial network, and one approach is using this Cytoscape open source software that, based on an abundance matrices, is possible to construct randomized networks that are generated by permutation for null distribution calculations. And then by bootstrapping, this is possible to assess the robustness and the statistical significance of these networks or the properties of the network. By analyzing these networks and visualizing them, for example, by Gephi, we have identified some hub taxa in these pre-alpine grasslands that belong to the proteobacteria and acidobacteria. These networks we have also analyzed at the ASV level, and what we see is that these hub taxa are not necessarily the most abundant ones in these grasslands, and also we have found that the positive associations between the grasslands decreased with the intensity of the management. So another possibility of what we have also implemented is using this software, NetComi. This software uses different methods for treating zeros in the data set, also different calculations for normalization and microbial associations and so on. And however, the output of these networks that is, you can get, for example, the summary about the size of the network, the cluster, the hubs, and also some centrality measurements. But this output could be different according to the parameters and the thresholds selected from the beginning, even at the taxa level. For example, here is with a high threshold, and here we get this with lower thresholds. So the shows, in general, there is no consensus to see which is the right way to estimate and analyze these microbial networks, and the choice should be made based on the research question before starting this analysis. Then if we want to see if these taxa are part of the core microbiome, one possibility is to use, for example, this AMPIS2 packaging R, which use two kind of thresholds, the abundance, or in this case, in how many samples are the OTU present and is considered as core. For example, by default should be in 80% of the samples, and the relative abundance of this OTU in the sample. However, this package is possible to adapt for when you have three categories. In our case, we wanted to compare these pre-alpine grasslands with another temperate grasslands, that is the Schwäbische Alb, in which we have used 25 plots that compound these grasslands and have been characterized by different land use intensity indexes, categorized like low, medium, and high. And in this case, altogether, we keep us 6 different categories that in a Venn diagram, that is the usual way to maybe visualize this core taxa, is a little bit more difficult to read. One alternative of visualization will be these upset plots like that, in which we can indicate, or it's possible to indicate, which are the shared taxa and the unique species in the pre-alpine and also in the Alp grasslands. And for example, in our case specifically, these 76 taxa are indeed shared taxa. We cannot say that these are really the core microbiome, because in this specific study, the taxa could be present in only one plot of these plots clustered in this specific category. So that is one point.
Diana,
Yeah.
just
Okay.
1 minute to remind you.
Okay.
Thank you.
Thank you. We need also to define if the taxa level. In this case, we work with the species level, and for that, of course, also the number of the taxa we will constrain, and in our case, we found that the taxa at species level was similar between pre-alpine grasslands and Alp grasslands in low amount, but the most number or most of these species were shared with the grasslands that were with low use intensity. And with this, I want to finish the presentation, and I will be happy to answer your questions. Thank you very much.
I saw that you said you don't define the core taxa based on how the shared similarities. So I think I missed it. What was the criteria you then used in really defining the core microbiota in your study? I'm just curious about this.
In this case, we define this core taxa when the OTU or the ASB was at least in 80% of the samples. That was our first threshold. I don't know if you...
Yes, I get the point, but I'm just a bit confused. Maybe this is just me, but if it's shared 80% in the samples, this is still making reference to shared taxa, right? Or where is the difference?
Yes, this is our reference in this case, but depend on the samples and depend on the questions that you have. Maybe you need to decrease or the reference to 65 or 70%, but this depends on what questions you want to, or what kind of taxa you want to cluster together.
Okay.
Dr. Rodrigo Lesdema Maru is lecturer at the Department of Bioengineering at Imperial College of London in the UK. And today he will talk about engineering microbial communities with defined application using synthetic biology.
Thanks, Pamela, for organizing the workshop and everything. It's very nice so far. So I'm going to switch from soil microbiomes and human or medicine-related microbiomes to another type of microbiomes that are the industrial microbiomes, and from natural microbial communities to synthetic microbial communities in the next 4.5 minutes. So starting by, my group is based at Imperial College, and we are interested in moving from a petroleum-based economy to a bio-based economy. And we do that with microorganisms that, as you know, are very important for plant performance, human health, but also they produce a variety of chemicals and metabolites that are important also for industry, such as biofuels, materials, food, chemicals, drugs, and so on. So while they produce these molecules, they produce them usually in very small amounts that don't make the process using biology feasible. So what we do is via synthetic biology, we engineer the metabolic pathways of certain microbes to produce this compound, that specific molecules of interest in higher yields. And what we are going to be talking about today is how we can also use not a single organism, but a microbial community that we design to maximize the production of some of these molecules of interest. So traditionally, this field of bioproduction, of metabolic engineering, is based on monocultures in one single cell, a clonal population of cells. And you can choose a variety of different organisms. And this is the one that we work with in my lab, but there are many people working with many other organisms around the world. We focus mainly on yeast, although we also have some projects with bacteria, cyanobacteria, and so on. And we work in a variety of products with different applications, from vitamins, fuels, nutraceuticals, flavors and fragrances, materials, proteins, etc. And we do this using different techniques that allow us to manipulate the metabolism. So we heard about metabolic models before. We use those metabolic models to identify what are the genes that we can manipulate to maximize the production of those metabolites of interest. We use techniques like multiplexing CRISPR that allow us to manipulate and overexpress and repress a variety of different genes in the genome in one single transformation. And we also develop methods to, for example, induce gene expression using like some fancy new methods like electricity. But something that we another have realized in the past years is that monocultures have limitations, and there's a lot of text in the screen. But the key points are that the more we engineer a microbe, the less robust it is, and the more easy it is to break that production capacity that we are trying to engineer, because they get overburdened. There is not enough resources to maintain their normal physiology if we push towards production only. And also some there's technical limitation that we don't know how to transfer like huge amounts of DNA into some organisms, and sometimes we don't know enough about the biology of certain pathways to be able to transfer those into the organism of interest. So we are kind of looking back to nature, and nature works in communities. There is no single organism that all the biological cycles and so on. So there are a community of organisms that are coordinated and more or less evolved to maximize their presence in certain conditions, ecosystems, and so on. So we are trying to bring back that concept into the industrial biotechnology. So we envision that the bioreactors in the future are going to have a community of organisms instead of a single yeast doing conversion of A to B. And we will be able to create certain behaviors that allow those communities to be more efficient than the monocultures. And of course, by doing that, we learn about microbial metabolite exchange, about relationships of cooperation, competition, etc. And we can apply that for other microbiomes. So we work in both what we call semi-synthetic microbial communities and fully synthetic microbial communities. The first one is when we take a microbial community and we engineer one of the members of the community, and fully synthetic microbial communities when we create it from scratch. So very briefly, in semi-synthetic microbial communities, which work in soy sauce, so it's a complex community of a lot of bacteria and some yeast. And what we've done is, because there was a company interested in that, reducing the dark color of soy sauce by identifying organisms that can reduce the components that are involved in the production of these melanoidins that contribute to this color. So moving quickly to fully synthetic community and what we are doing there.
Rodrigo, just 1 minute. Thank you.
Perfect. Yeah, we work with a variety of different communities, and we are mainly interested in 3 behaviors that we want to engineer and control: division of labor, so separating pathways into different organisms; cross-dependencies on syntrophy, exchange of the metabolites that are necessary for the other strain; and population control. Because if we control these 3, we can design this type of bioprocesses that I mentioned before. And just briefly some examples of what we've done. So we've produced raspberry ketone, so the fragrance of the raspberries, by creating communities of 2 or 3 yeast that exchange metabolites, and they do it in relatively high proportions. We've also engineered a division of labor for malonic semialdehyde together with syntrophy and population control with different ratios, and found that communities work better and produce twice as much as monocultures. And we've recently also demonstrated the same for resveratrol production with an antioxidant, combining division of labor, syntrophy, and population control, population dynamic regulation. We can produce more with the communities than with single organisms. And with that, I thanks everyone in my group and all the funders and all of you for your attention.
Tim?
Yes, hi. Thanks for that great presentation. I was like wondering, so are you currently using metabolic modeling approaches for generating and defining the synthetic microbiome communities? And if so, how? Like, is it, yeah, just
A great
curious.
question. So we haven't, the metabolic models, we are only using them for the moment for monocultures. We done a quick test with co-cultures, but it's not also the core expertise of the lab. So yeah, we will be happy to explore any collaboration in the metabolic modeling of community side of things.
Yeah, indeed, it would be really interesting, I think, to have a digital twin of a synthetic community, which can then iteratively enhance each other. Yeah, thanks.
Thanks.
Let's welcome the first speaker of our second block. So she's Professor Lisa Meyer. She's full professor of microbiome host interaction at University of Tübingen in Germany. Her research group uses interdisciplinary approaches at the intersection of microbiology system and computational biology to study the human microbiome. So today she will talk about opportunities, challenges, and pitfalls of microbial model communities, insight from in vitro and in vivo studies.
Okay, thank you very much for the kind introduction. Thank you very much for the invitation to speak here. Yeah, so now we switch a little bit topics. We are going to the human gut microbiome. And yeah, it's my great pleasure. It's been fantastic to prepare for this workshop, because actually I've been going back 10 years and how we selected our core species, and now see the tremendous possibilities that you have to do, that you have now. So yeah, I hope that from my talk you can learn a little bit what we did, the amount of additional work this created, we've done, what we have learned from the way that we've done it. We've selected our core species and our keystone species, and I hope this will be informative for you as well. Okay. So I'm interested in the human gut microbiome, and back then, and that is now really going back to the beginning of my postdoc times at EMBL in Heidelberg. So we are talking 2013. It became clear that maybe not only antibiotics can change the composition of the microbiome or can influence the composition of the microbiome, but also other drugs. And there was this one poster boy example, which was metformin. And we asked ourselves, so, I mean, if this is one drug, non-antibiotic drug, that can impact the composition of the microbiome, is there also maybe other drugs that can impact the composition of the gut microbiome? And can we assess that in a direct, systematic way? So cohort studies are great. We've learned a lot, and really, I love them. However, if you want to understand the causal relationship, it can be really helpful to take a bottom-up approach. And for us, our solution was a cultivation-based study. So what we set up in the last 10 years now is really like a bottom-up approach for cultivation-based microbiome research for the human gut microbiome. And what we do in my lab, we usually start with investigating interactions, drug microbiome interactions in pure culture, and then we stepwise increase complexity. So when we have mapped a direct interaction, we map these interactions in communities. Then in stool-derived communities, we increase readouts on the host side, so host cell readouts, and then complement those with studies in chnotobiotic mice, so mice that are germ-free and then colonized with a certain set of microbes under very controlled conditions, and then compare to population studies. And we have exactly that problem that has been raised many times before. Yeah, I mean, it's many, many species to look into. And we want to understand a variety of different drugs. So we have to prioritize. So in our case, we try to automate as much as possible. So we work in anaerobic chambers to accommodate the needs of the anaerobic gut bacteria. And then we do scalable and automatable assays, have a variety of different assay designs. In many cases, we just look at growth phenotypes, but we have also set up reporter assays, germination assays, slices, relative or absolute abundances, and microscopy for certain drugs. We also do multi-omic studies. And we've successfully used that setup in the past to map the effect of non-antibiotics and antibiotics on the gut microbiome. But in collaborations with others, we also looked into other stressors like salt, bile acids, host adaptation, a way to have antibiotics that are more microbiome friendly. And we've put everything together in a comprehensive protocol on how we do things that has been published last month. So this is available. And if you have questions, please always reach out. Okay, so as I mentioned, we had to prioritize. Microbiome's complex, and the challenge was to define which organisms to focus on. Now, as mentioned in the beginning, so this is a problem that we had in 2012, 2013 for our case, where we wanted to map the direct effects of drugs on the gut microbiome. And the problem was basically, it's pretty clear that the drugs have an effect. We knew that from cohort studies, but we wanted to understand, is this a direct effect? Would these non-antibiotic drugs directly inhibit members of the gut microbiome, or is this more host-mediated effect? And it's not only the direct inhibition or boost of individual members of the gut microbiome, but it's also which one could bioaccumulate or degrade certain drugs. And therefore, we wanted to have the systematic map. And I mean, to do this in a culture-based approach, we had to select members of the gut microbiome that we wanted to study. And our selection is shown here. So what we did is we tested the effects of 1,200 FDA-approved drugs on 40 members of the healthy human gut microbiome. So that's 40 strains, 38 species, and 21 different genera that covers the healthy human gut microbiome. And that is really, at the time in 2013, 80% of the assignable average abundance at the genus level and 60% at the species level. And we then did this high throughput screens. And I have to say that, mapping all these was basically 3 to 4 years of experimental work. So you really want to wisely choose the microbes that you study. So at that time, we had help from Georg Zeller at EMBL in Heidelberg at that time. And what he did, he looked into, I think, roughly 400 metagenomes. That was what was available at that time. And this was from cohorts across 3 different continents. So we had Spain and Denmark, the US, and from China. And he looked into taxa that were present with an abundance of more than 1%, at least one sample, and 50% abundance across all the samples that we have tested. And then he defined a core of 60 taxa Now this is represented here at a general level and how much species we have in the different taxa. And we basically ranked that. So that's the genus level. Now this is here the species level. So this is basically the list that we got from Georg. And what we did is to find out whether we can find cultivable isolates, which was also not so common and easy to find at that time. And everything that is marked here in gray is actually things that we kind of included in our studies. So in the end, we had 40 different taxa, 38 different species that we sought out a core microbiome of the healthy human gut microbiome. And then, I mean, we had to select always like one isolate. So we decided to go for type strains. And this is what we did. We did this gigantic screen, yeah, and we learned a lot. I mean, tons of work, but we learned a lot. Of course, antibiotics have a direct impact on gut microbes. We have now a systematic map, at least for the type strains. That's even for the antibiotics, that was new because we had a good understanding on their direct effects on pathogens, but not necessarily on commensal gut bacteria. We also found that for many non-antibiotic drugs, so human-targeted drugs, as we call them, we find a direct effect, at least at one of the species that we selected within our selection. So we've identified more than 200 drugs that directly inhibited gut microbes. And they were across all therapeutic classes, but enriched for the calcium channel blockers, for the antipsychotics, and for the antimetabolites. And that raises, of course, a lot of questions. The antipsychotics, I mean, they would target transporters or receptors that are not necessarily present in bacteria. So this is a lot to follow up on, to understand. And for the antimetabolites, it makes sense. It's basically conserved pathways between pro and eukaryotic cells. So this is a gigantic data set. It's really helpful for a lot of different disciplines. I mean, this paper is really highly cited because it raised so many new insights. So I mentioned it because I really want to emphasize that having these culture-based approaches can be very fruitful for a field. Yeah. But for us, we wanted to go ahead. Yeah, I mean, this sensitivities in monoculture, that's one thing. But in reality, we have complex communities that colonize our guts. So we wanted to take the next step and also look into defined communities and also complex communities from stool samples. So we basically kind of expanded our protocol for communities. So in the very first step, what we did, so I mean, we had, as I mentioned, we had a lot of work done in mapping the direct effects. So we wanted to know for exactly those microbes, how would they behave in a community? So we took the 40 that we have, looked on what we can distinguish by 16S RNA sequencing, also to make follow-up experiments cheaper, and then basically identified those that we could distinguish by 16S sequencing. So these are the 30 species that I've highlighted here. And we then put them together in exactly the same gut mimetic medium and try to understand whether we can recover all of them, whether they would kind of form a stable community. And this is basically indicated here. And what you can see is the relative abundance. And this is just a fresh culture if you inoculate in a 1-to-1-to-1 ratio, all of them. So we did a lot of experiments on whether we need a 1-to-1 ratio, we can do another ratio. And to basically sum up a lot of experiments, I think what I would really like to emphasize. So in the end, what happens, we have like 31 species that are more abundant, so the top ones here. And even if we don't add the less abundant ones, it forms the same kind of community. So here's a little bit an experimental problem. But in the end, what we know now, if you put them 1-to-1-to-1 together, in equal ratios, we always have this community of this particular composition. We can do this from glycerol stocks. We can do this in several passages over time. I've had a lot of students having to put this community together, and we always have this one stable community. So this is quite helpful for us. And we can do the corresponding follow-up experiments. However, it also says that there is quite some intrinsic stability. And what we do now is basically we ignore those that we always pilot but never find back. We also have now procedures available in the lab or in a university hospital here where we collect complex fecal samples. Here the key is really to put them under anaerobic conditions as fast as possible. So we do this here with a kit. The kit is super expensive. We could do it in a way cheaper way, but the clinicians really like having kits and something with clear instructions and not something experimental that comes from my lab. So we have made good experience in engaging also clinicians to help us collect samples using this kit. And here it's also pretty clear. You start with a higher diversity or a species richness, and then there is a drop as soon as you put them in cultivation. But then it doesn't matter too much whether we take a cultivation from a fresh sample, from a cultivated frozen stool sample. If we put new stocks from the stocks that we already had, it's basically always the same composition. So it's really the environmental conditions that basically drive the composition of the community in our case. But now, I mean, for us, it's now perfect. We have the experimental conditions that we wished for. So we can take these species, these 21 species in monoculture, expose them to drugs and look at the relative growth. And we can compare it to our community setup, where we have here there's 32 species, but basically it's 21 different species, have exactly the same drugs, incubate them and look at the fitness and the absolute abundances, and then compare what happens in monoculture, what happens in communities. And this is now work from Sarella Garcia that I want to present. So in the end, what happens, and this is already mentioned in the talk before, communities are more robust. So we see a protection in a community setup. So quite a number of species are more protected in the community, in particular at low drug concentration. If we increase the drug concentration after a certain dosage, basically this protection breaks and less species are protected. And this is mainly because of bioaccumulation. So certain species would accumulate the drug, or biodegradation. So they degrade the drugs and others are protected. We can also look into cross-sensitization. And what you can appreciate here is that this is roughly 6 times less frequent than the cross-protection. And what you can also observe is that this actually increases with higher concentration. So that's basically what we learned from those kind of experiments. And I think it really matters to look into what happens actually in a community. Now for us, we want to also go the next step ahead, and that's basically looking at exactly the same communities where we have drug sensitivities in monoculture and in the community setting, now in the host setting, and therefore we colonize germ-free mice with our communities. So here is a little bit the setup. So we have this isocage system where each cage basically is like a completely sterile environment where you have the mice in. It's like a HEPA filter system where we have like the possibility to have mice with different colonization here in the system. So when we kind of handle these mice, we put them here in the cages in the dung tanks. It's completely kind of filled with disinfectant, and then we can, from the inside, open here this tank and then work with the mice. And here on that side, we can put in all the sterile equipment that we need for handling. And this is now what we did. So we took our in vitro COM30 community and colonized germ-free Black 6 mice. So this is the first colonization. We also repeat the colonization, so in case something went wrong, so that we make sure that we do it actually twice. And then we do fecal sample collection over time and determine what's the composition of the microbiome. So this is what we in the end do. We take a last fecal sample and then do a cultivation step again in the same culture medium as we had our in vitro communities. And I guess what you can really see from the COM30 experiment is that there is a big difference what we get out in what we actually put in. So
Lisa, so 5 minutes just to
5 minutes, that's perfect.
Thank you.
Thank you. So this is very different from what we put in. And then we feel like it's difficult to compare drug sensitivities. It's also kind of a quite complex community that we have to put in, so it's quite work as well. And I want to emphasize here that I mean, this is important information for us, that we have different compositions in vitro and in vivo. We did the same thing with COM20. So those that we also know that they can stably grow in our in vitro conditions. And again, did exactly basically the same experiment. And here you see that we have quite a good comparison between the in vitro and the in vivo system in that community. So now we can also look back into, all the metagenomics samples that have been sequenced. And now across more than, I think, 5,000 different individuals that we have metagenomic data for now, we can still say that roughly on the metabolic pathways that are represented in our COM20 communities, we cover, I think, more than 65%. So we have quite a good coverage, both in vitro and in vivo. This is just another way to plot the data. So basically, this is our COM20 community. This is our original inoculum. This is when we have like the culture from fresh or from a frozen stock. It's basically the same. And then what can colonize mice for a really long time. So for us, we have now basically the perfect setup that we can do this comparison of what drug effects are really community mediated, direct drug effects, or host drug effects. And now we have the first experiments running where we can actually look across scales the drug microbiome interactions. Looking ahead, we also do bioreactors to, in the long term, also kind of decrease the amount of mice that we would need, and also for evolutionary experiments. So our setup at the moment is mainly this 96 well blade, so that we can really have the throughput that we need to investigate the different drugs. But we have now for certain drugs the first models where we go on gut on a chip and organoid models, in particular, to model now gut-brain axis signaling. So opportunities, I think this is what we have in hand, is quite a cool model. However, it also took us 10 years. So from selection of what we think is the core microbiome to what we do now happened a lot, also in terms of the diversity that we understand. We basically kind of try to overcome this by looking also in the individualized, like complex microbiome from stool samples. And then there is a lot to do. We can integrate other, not only bacteria, but also archaea, fungi, and so on. And still for the complex communities, and this is something I would also happy to get a little help and ideas from this crowd here, is how to cryopreserve complex communities and what's your experience, because we've done this now for 10 years. And we have some experience, and not all of the things that we actually tried work, and they're having like a sustainable way would be also something that I find would be important for the community. Our model, of course, has some pitfalls, so it's really on drug microbiome interactions. We know that certain traits of the microbiome are not really nicely reflected. It's a mouse-human hybrid, so we have human isolates that we have in mice, so that's maybe not best if you want to look into immune system responses and so on. Still, it has a low complexity, but we hope still to kind of reflect many of the things that we want to study. Yeah, and I think this is basically what I wanted to share with you from our experiences, and I'm very looking forward to see where the field goes, and the idea is to maybe do this better in the future, and maybe also to improve our model. Thank you very much for your attention. I think Michael was a little faster.
Then Michael.
Yeah, thanks, Lisa, for this really fantastic presentation. I really enjoyed it a lot. What is the influence of diet? So when you do your mice experiments, of course, it changes the community, but it can also change the bacterial phenotype. So can you distinguish what is the response of the change in the community and what is the response of a particular phenotype, meaning that you need certain amounts of nitrogen and phosphorus, that certain genes are expressed and so on. And this might also then determine a little bit how resistant an organism becomes against antibiotics.
I mean, definitely, very much looking forward to look into the diet component at some point. We are not there yet. So at the moment, we use standard mouse diet. We are happy to change that at some point. And I did not mention the medium that we take for growing our in vitro communities. We tested, I think, 20 different media, and then looked at the composition and used the gut microbiome composition that more resembles the in vivo conditions. So we feel like our medium our in vitro medium is well chosen across different possibilities. So we are happy, and we see similar compositions in the mouse gut. So we think that makes sense. However, when we have like a high-fat diet and more bile acid, and I have the bile acids in combination with an additional drug, that might screw up everything that we've tested before. And that's something that we have to investigate in the future. And that's also our plan. So yeah, I'm very curious to see, because if you can change how the drugs actually work depending on your diet, I mean, that's huge potential.
Hi, Lisa. Thanks very much for the presentation. So would you say that so there are some things that are known to negatively correlate. So in the gut microbiome, Enterobacteriaceae negatively correlate with Christiansen Eller. I think on one of your other slides, when you were also doing the passaging, I think you lost a Prevotella. Is that correct? There was Prevotella, and then suddenly it dropped out.
I'm sorry, that was really an experimental mistake. So I wasn't that was not lost. That we know that was a problem with the experiment.
But for these
I'm sorry, but normally that works. Yeah, Prevotella is always a little tricky, in particular for mice. But we think we can handle that. But that's
okay.
Human mistake, not an observation. I'm sorry.
But for these things that negatively correlate, do you think that the way to establish that understanding of the mechanisms is to try and, would you see in the competitor once you've got it at a stable community, or is this something where you'd actually have to establish the two separate communities to try and understand the community dynamics of what, I'm sort of trying to understand. So you talked about stabilizing the community. I'm sort of now thinking about the next step of how you start looking at the dynamics of, if you start introducing competitors that we know negatively correlate.
Yeah, so here the question is, at least for me, can you do this in a one-to-one interaction, or would you need then the community? And can you then restable within the community? Because I think it's thing where you have like a direct warfare between two species. Yeah, I mean, this can be done in high throughput assays as well, maybe on a one-to-one level. If this then is a community phenotype, I guess the way that we build for the drug microbiome interaction, this is the way that you would also go. Yeah, you have the communities, you have the possibility to either drop one out or add one on top, or two or three, or sub-consortia. At least that would be my bottom-up approach to the question.
Okay, all right, thank you.
So let's try one more quick question, Marcus.
Yeah, just a quick question. We are doing the farm children studies, and we see actually also the impact of the antibiotics on the gut microbiome in the first year of life. And we see also the impact of the nutrition, breastfeeding and nutrition, but we also see impacts of the outer environment. So a transfer from the outer environment to the gut microbiome. Do you think that you can model this also in your approach?
Yeah, that we continuously feed with, so this is basically either the batch culture or the continuous dilution into new 96 well plates. That's basically would be our approach. And then we would always add, an incoming microbe. And I have not talked a lot about that at all, but we do a lot in terms of colonization resistance. And I mean, colonization resistance is like the classical way is like a pathogen that would invade and grow in a community. That could also be like a probiotic or a bacteria that is basically coming from the environment. So we have ways to tag them and then monitor them in the community so that we don't have to sequence the whole time. Also in high throughput, because again, we want to investigate this across a variety of different conditions. So this is things that we think along, mainly now for the pathogens, because we feel like is like the lowest hanging fruit, because the question is if non-antibiotic drugs would actually impact the composition of the gut microbiome, would they also kind of disturb the microbiome and allow pathogens then to colonize? And if this is the case, I mean, that in particular for, old people that take many, many drugs, that might be important to know, and maybe even possible to assess whether colonization resistance is broken. And this is how we think, but that could be implemented for any incoming microbe within a community.
Also for the beneficial ones.
Or the beneficial ones, or, environmental sampling.
Okay, yeah, thank you.
Our next speaker is Marco Fabrini. He is a PhD student at a unit of microbiome science and biotechnology of the University of Bologna in Italy. He is part of the group led by Professor Silvia Turroni, whose research is focused on the study of microbiome host coevolutionary trajectories and the impact on health and disease, food production, environmental health, among others. So today, Margot will talk about microbial networks and keystone taxa in human health and disease.
Okay, thank you so much for the introduction. I hope you can hear me and see my screen. And today I'll be presenting a brief overview of microbial network and keystone taxa. I know there's been a lot of discussion on this topic already, so I hope it doesn't get too repetitive for you. Taking Banerjee's definition of keystone species, they actually can be defined as highly connected taxa that in a guild exert considerable influence in the microbiome structure, and they have unique and crucial role in the communities, and their removal cause a dramatic shift in microbiome structure and functioning. So several methods have been proposed for the identification of keystone species even today, including differential analysis, so present, not present, network-based models, we've been discussing them a lot, and also genome scale metabolic model and perturbation methods, as we already explored today. So we already saw a couple of networks in the different presentation, but I would like to give you just a brief repetition of the logic. So networks are a set of interconnected elements which are visualized as nodes, so the entities connected by edges, the connections. And the network theory mostly developed under like the social network analysis, where it demonstrates great potential in modeling patterns of communication and in mutualistic influence. So it's been adapted to microbial ecology in describing the relationship between the members of the microbial communities as interactions or association values derived from several methods, mostly correlation, co-occurrence, or different heuristic, starting from compositional data to describe some complex interaction in the form of networks that we can analyze, evaluate, and from that gather some insight on our community. So the network theory adaptability to microbial ecology is also underscored by the potential increase in article employing the term microbial network in the recent year. So this surge reflects the growing recognition of network theory efficacy in unraveling the complex relationship among the microorganisms. So networking approaches mostly rely on observing the ecosystem as is, mostly from compositional data, but it can be done also at different level. For example, observing the functional potential in terms of pathway abundances, for example, also adding also like metabolic concentration or all of them. So constructing microbial networks nowadays primarily relies on applying co-occurrence or correlation or association methods, as we've told, on such compositional or functional or metabolic values and derive these links between the members. So the premise on that is that specific organization of the ecosystem that we can observe from this with these techniques hint underlying biological interaction which is happening between the member, which can also include nutritional interaction. So the methodology offer a lens into the unseen relationship among microbial community members, which can help like enrich our understanding of the ecological dynamics happening. We need to characterize the network to gather some interesting insights from them. So the first thing we can do, we can employ topological measure, so like more technical approach to describe the ecological intricacies. So the first thing we can do is evaluate modularity, which involves taking a network and divide it into like modules or communities, which can reveal some subgroup of microorganisms that we can say frequently interact among them. And we can use different algorithms to do that. And then we can observe centrality. So centrality measures, which mostly include degree, closeness, betweenness, eigenvector, are a method to describe like nodes and score them in like a degree of importance based on different parameters. So for example, the degree centrality evaluates just the number of connection a node has, Betweenness highlights the node at the interface between that bridges like different communities in the network. Closeness evaluates how quickly a node can reach the others, and so on. So we have different measures to highlight different properties of network. But aside the topological aspect of the network, so simply evaluated the network structure, we can also use other measures of connectivity. And for example, here we like to just briefly show you the cohesion, which is a measure introduced by Heron and colleagues in 2017. And it's simply can be like simplified in a matrix of average correlation values between all the microbial members. So the fact is that this measure can be observed both in terms of positive and negative values, so focusing on likely beneficial or competitive relationship. We can represent it as a ratio, and we can observe it as a whole, just to give a holistic view of the community. And an interesting work presented by Hernandez and colleague in 2021 evaluated the soil microbial diversity and composition using this parameter, and they observed that in case of lower stress from like hydrical and nutrient stress in the soil, the richness was higher, the network was more modular, so it was strongly divided into separate communities within the network. So the ecosystem was allowed to have more competitive interaction. So the negative cohesion was higher, and a stressor was likely to be propagated only on few members of similar on the same module of the impacted one. Whereas in case of higher stress, the richness is lower, the modularity is lower, so the community is forced to rely only on positive association because the negative ones are wiped out by the stress, and an external stressor is likely to propagate on the community. So we thought that this parameter was of great interest, and we used it in the way that we defined in one of our work, the keystone. So what we noted is that topological analysis can measure hub nodes, so highly connected nodes with large number of interaction depending on them. But actually, we thought it was not like in the real scope of the concept of keystones, as we saw in the first slides. So as presented before, like for example, by Dr. Vieira and others, we use the leave one out approach as well on microbial networks to like define keystones. And it was, I think, first presented also by Barry and colleagues in 2014. They did it on simulated community. So basically, we applied this approach in a cohort of human gut samples. We analyzed with shotgun metagenomics of pediatric patients undergoing hematopoietic stem cell transplantation. And we decided to determine keystones in the human gut microbiome as the species whose removal, like with the leave one out approach, caused the most significant reduction in total cohesion. And looking at one of the networks that we reconstructed, and this table of the taxa that were detected as being hub for the centrality measure and being keystone with this leave one out approach, you can see that there are some taxa which are shared, like the two underlined here, Faecalibacterium and Nigritella, which likely makes sense also based on the trajectory observed for this patient and their health status and their response to the therapy and so on. But what we saw is that using only centrality measure often identify hubs based on just the presence of a lot of connection or one strong connection, whilst the use of keystone identification with this cohesion, and cohesion takes into account also the relative abundance, leads to like the identification of keystone based also on their relative abundance. So for example, these two Bifidobacterium strains likely represent a huge fraction of the community. So their removal is likely to do something to the ecosystem. So we thought that combining the output of the two approaches would be like good. And indeed, we got some interesting results that strongly associate with the clinical phenotype. So we decided to use total cohesion as a metric, but evaluating multiple parameters is arguably the best and more robust choice. And what we noted is that not hub or all hub nodes are keystones and vice versa, because like definition is different, the method they are calculated is different, but they both associate with some clinical features, so they both seem to be meaningful. And we already saw today lots of articles discussing about this, but this is just one I would like to introduce you just for they use leave one out approaches on NGS data. So it's something that has already been done, as it's been also presented today. We must acknowledge some limitation of this approach. First, we have a vast range of techniques that we can use to model microbial communities into networks. We cited co-occurrence, correlation networks, but there are also several tools like Speciesi, Netcomi. They use different heuristic. The results sometimes are quite different. So this can introduce variability in result. And in addition, we lack a functional representation of the metabolic interaction. We must focus on bacteria. Lots of studies are coming up with like focuses on viruses and fungi, but our ability in defining them, I think, is a little bit lower than the focus we have on bacteria. And unlike hubs, which are like well recognized mostly because of other science field approaches and social network theory, they are defined like using centrality measure, and mostly everyone agrees on that. The way we define keystone, as we saw today, lacks like a consolidated approach, so this can be also a limiting factor. And just briefly talking about the core microbiota, the definition of core microbiota has been a challenging task pursued by many in these years with different approaches. In our review, we highlighted some of them, like using just composition, integrating also the functional profile. Each one of them has pros and cons. But in light of what has been discussed today, in the light of this keystones taxa identification, like if we detect consistently concordant keystones across very large cohorts, we also might use them to help us define a core microbiota. So we can also consider this approach as feasible for studying a core microbiota. I think that approach integration would be the best way to go on, keeping in mind that we have strong biases in defining core microbiota because we mostly focus on genomic data, although it has been, we saw some very interesting approaches with culture-based methods today. The data is mostly Western focus, fecal-based, and we have lots of amplicon studies, not a lot of metagenomics, but we're moving on. So I just want to conclude so that networking approaches I think could be useful in deciphering this complex interaction. And we used leave one out approaches on total cohesion to identify keystone, although multiple parameterization I think could be the best way to go into this field. And there's a lot of things to do. There are a lot of limitations in these approaches we need to acknowledge, but I think it's a possible way that we can use to define the core microbiota. I've been quite short, but I didn't want it to be repetitive, so I thank you for your attention, and I'm open to questions.
Hi. First of all, thank you for this excellent overview. So I was very interested, especially in the cohesion metric. But I was wondering, to calculate the cohesion, you use the expected correlations. Like, how do you define expected correlations?
Yeah, actually, we did not use expected correlation. We like calculated the correlation like between, as proposed by Heron, between the different taxa in different samples. So we take relative abundance and use the correlation values, and we also did it with mere correlation like Spearman, Pearson, and so on, but also with different association values derived from several approaches like SparseC or SpeakEasy. So once you have a value that can be either positive or negative, linking all the taxa among them, this approach can be pursued. So we don't calculate anything expected. We just use the data and take the data as it is.
Thanks.
Yeah, Marco, thank you very much for your very nice overview on all these methods. I do have a question to your multi-parameterization approach. I remember papers which started off with a Lotka-Volterra simulation of communities, so they know which guys have what type of interactions and which are keystones. From that, they simulated abundance tables. And these abundance tables, they then kicked again into network approaches to identify those keystones again. And I remember the effectiveness of the statistical identification of keystoneness was not the very best. From your experience and from your overview, would this multi-parameterization approach be better? Once you know you have a community and once you know already a priori which are the keystones?
It might be. Maybe the approach is not feasible for keystone identification. I'm not saying it's the only way we can proceed, but I think that adding more parameters into play, especially because when dealing with communities which can interact at different level, if we only consider this relative abundances, I think we are lacking a lot of the possible interaction, indirect interaction that can actually occur in the community. So adding metabolites and maybe in the future, like metabolic models that can take into account how a metabolite is affecting different taxa is something that maybe can help this approach get really the robustness that we need to actually consider it strong and feasible. So right now, I agree with you. We encountered some like fluctuation in these approaches. So they're not like bulletproof, but I think they have some potential. So I think, yeah, multiple parameterization might be one way to.
Professor Vanessa Stadbauer is full professor of translational microbiome research and hepatology at the Medical University of Graz and CB Med in Austria. Today, she will talk about the core human microbiome. Does it exist?
Yeah, thank you very much. I didn't have the opportunity to follow the whole meeting today, so hopefully I am not too repetitive. And I was asked to provide my view on the topic, if there is at all a core human microbiome, and all of this within a few minutes. So I'll do my best to meet this task. So I will give a very brief overview on the development and composition of the human gut microbiome, and then try to answer the question if there is a core or keystone human gut microbiome, and touch briefly upon the topic of human gut microbial diversity. Yeah, so as you probably already discussed at some point today, you know that the human gut microbiome harbors a lot of microbial cells, 10 to 100 trillion cells, also a lot of genes, more genes and more cells than in the human body, and that 90% of the phylotypes in the colon belong to two bacterial phyla, Bacteroidetes and Firmicutes. However, as opposed to the composition of the human genome, the human gut microbiome shows a large variation between individuals. And it's maybe something even you can think about it as a philosophical question. Why is there such a high inter-individual variability, and how is this sustained? I like this neutral theory from Stephen Hubble a lot, who says that in such a complex ecosystem, it's rather the function that is common between the individuals than the actual composition of this ecosystem. When we look at the development of the human gut microbiome, we know nowadays there is still some discussion ongoing if the fetus is already colonized in the uterus, probably not to a large extent, only maybe with a very low number of bacteria. And then at birth, depending on the birth mode, there is the first colonization with vaginal delivery with lactobacilli, and with cesarean delivery mainly with bacteria that can be found on the skin. Then during the first 1 to 2 years of life, the microbiome develops, depending also on the type of nutrition a baby receives. When solid food is introduced and the babies grow up to toddlers and then eat a diet resembling a full adult's diet, then we find a microbiome that is also comparable in its composition and especially in function to the adult microbiome. There are a lot of influencing factors like drugs that are taken in these first very vulnerable 1 to 2 years. As I already mentioned, the mode of feeding and also the environment that shape the microbiome. Although the microbiome between babies with vaginal delivery and cesarean delivery look very much different, and this difference can also be seen several years after birth, until now, it has not been proven that it makes a difference to health in general or also to certain diseases if your first colonizers are lactobacilli or if your first colonizers were more from a bacteria from the skin. So explaining this extreme variability in the taxonomic composition is not so easy. There are several factors, of course, that have been identified. That's host lifestyle and diet, of course, as a main factor. Also, drugs that are taken by the host genotype, the host immune system, the presence of transient community members, and the environment where the host is in. When looking at the functions, it seems that there is much more homology between individuals. So this was one of the first studies, I think, that was done with metagenome sequencing, where also human gut samples were included. And it has been shown there that the functions that could be found contribute to things like energy harvest, metabolism of xenobiotics, organ function and renewal, immune function, and behavior.
Vanessa, I have
Yes.
1 minute just to give you a feeling.
Yes.
Thank you.
Then you probably all know this concept of resilience, that a highly diverse ecosystem is more resilient to damage from the outside. And I think this is a key thing in the human gut microbiome. So it's not so much the presence of individual strains. And when we just look here on the right side on this graph, that tried to combine different culture-dependent and culture-independent studies on the human gut microbiome to find overlaps. You see that there is actually not a lot of overlap that stays consistent, like in the middle of this graph. So it's more the diversity and the functions that we have that determine the health of a microbiome. So in disease, we have this concept of dysbiosis that is often associated with a reduction in diversity and a reduction in resilience to changes from the outside. And I would just like to come to an end of this talk, then show you an example from one of the diseases I study quite frequently. This is liver cirrhosis. In liver cirrhosis, we see this really severe loss in diversity and this difference, so in alpha diversity and this difference in beta diversity. And we could also identify that intake of certain drugs drives this dysbiosis. For example, when you take in stomach acid blockers, then you get an increase in bacteria that usually come from the oral cavity in your stool microbiome. And this is then associated with inflammation and a leaky gut, showing a loss of colonization resistance to other pathogens, and also a deterioration of the disease, and even a worse outcome in the sense of a 4-fold increased mortality over 3 to 4 years of time. And just one sentence to the end: diversity is not always the key. There are other body regions, like for example, the vaginal microbiome, where a low diversity and a dominant lactobacillus strain is the key to health, whereas a high diversity is associated with a higher risk of sexual transmitted infections, preterm birth, or bacterial vaginosis. So to summarize, I don't think that it will be possible for human health to define a set of keystone taxa because we have to look at the functions as well. The challenges are that it is very difficult to use in research, as we already heard in the previous talks. So I believe that we also have to link metabolomics data to our research to find out which functions are important, and then find bacterial taxa that can change functions rather than change the setup, the anatomical setup of the microbiome. Thank you very much.
Yeah, thanks, Vanessa, for this really nice presentation. You mentioned, if I understood you correctly, that the influence of strains is not so important. Taking into account the classical taxonomic definition of strains, this is quite surprising because it says 75 DNA homology represents one species. So there is 25% differences in the genome of two strains, and then it's still the same species. So you have any idea how to explain this discrepancy? Because I thought always that our resolution that we have with all these molecular techniques is simply not enough to cover all these strain-specific issues. And this is one of the problems why it's hard to predict functionalities.
Yeah, so this is of course so what you mentioned when I was preparing this, I was also thinking of how should I phrase this. Yes, I think that also our techniques are not, especially also for looking at human data, are not good enough, because then we also talk always about stool samples. Stool samples are very heterogeneous. Taking tissue samples, looking at the microbiome that really interacts with the body is always difficult. But I also think that there could be in human health, when we look, what's the role of this high diversity? Why do we have this high diversity? This must have a sense. Otherwise, we would have also in the gut microbiome only a few dominating species, like we have it with one dominating species in the vaginal microbiome. So I think that species could also, or strains could replace each other also functionally. So, and when we then think of therapy, we now struggle a lot. We have probiotic bacteria that have a long tradition in food industry, but only have a very defined set of functions. And we know that other bacteria that are present in the gut microbiome have other sets of functions. So how can we bring these functions that we would need in the microbiome into our body? I don't think that we have to focus on one strain, or also not on one species that has these functions. There could be several options, and that's still the problem, because that's also what we lack in regulation. In regulation, the current regulation doesn't really take into account that we have living organisms, the bacteria living organisms that could, depending on their environment, exhibit different functions. So yeah, I think that's not a really conclusive answer to this now, but I think there are so many things that have to be discussed and tackled, and researchers have to focus on one strain or one species, but also take into account that we could have an alternative.
So Juliet is PhD student at the Department of Comparative Microbiome Analysis in Helmholtz-Zentrum Munich in Germany. And today she will talk about community assembly analysis.
Awesome. Thank you, Pamela, for the introduction. So let's put this on presentation. Wait, sorry. Not right. Okay. So yeah, as Pamela said, I'm going to talk about community assembly analysis, specifically the theory behind it and also the application. So basically what we're looking at with community assembly analysis are what are the selective pressures that can influence the changes in population. There are two major theories of community assembly. One is a niche-based theory, meaning it's deterministic or there's some kind of selection happening. So on the left-hand side, you can see there's a species pool that has A, D, H, G, all representing different organisms, and then they pass through a filter and you get the same species at the end, depending on some kind of selective pressure. So that can be an environmental pressure like pH, temperature, moisture, salinity, or a biological pressure like competition, mutualism, predation, etc. And on the right side, we have neutral theory, which is stochastic, meaning it's randomly selected. So you can see the species pool up top, and then it goes through some kind of filter, which is a random filter, and selects for a random assortment of microorganisms, or organisms in this case. That all of the species, this assumes all the species are ecologically equivalent and that there is no ranking between them. So what I want to direct you guys to today is an alternative method to look at communities, which is this community assembly analysis. And it uses phylogenetic algorithms to assign and quantify specifically how much these processes are contributing to specific communities. So that would be selection, speciation, diversification, or stochastic things like dispersal and drift. So one of these algorithms is called beta nearest taxon index. And as I said previously, we can quantify the contributions of the assembly processes with things like beta NTI. Basically how beta NTI works, I won't go into the specific details. You can read James Stegan's paper from 2012 or 2013. He kind of invented this method. It has to do with putting together a overall phylogenetic tree and then looking at pairwise comparisons of other phylogenetic trees. And it has to do with shuffling the tips to see the correctness of each tree and comparing them against null models. And then in this method, it assumes more closely related taxa are similar ecologically and uses mean nearest taxon distance to quantify the phylogenetic distances between species in two communities. So you do this for every single comparison of interest, and then you get 999 null distributions, and you compare that against what you actually got. And then it gives you numbers depending on what are the percentages of each process. So an example of this would be a recent publication from my group that I headed was looking at biocrests, which are these organization of microbes that occur on the soil surface that have a lot of very interesting and potentially useful properties that are composed of bacteria, algae, fungi sometimes. And we looked at them on salt heaps that were remnants from potash mining. And so we sampled along the heap and we found just the pure salt heap initial biocrusts and then established biocrusts. And we found in two different sites that were 50 kilometers apart from each other that there were more deterministic processes that were dominating in the biocrusts at the end of this salt heap chronosequence. And that in the beginning, in the heap and initial phases, there were more undominated, or in this case, drift. That's what's classified as undominated in the white. And we saw that there was more variable selection, meaning the communities, there was some kind of selective pressure on them in the biocrusts. So why do we care about this? Many other publications have used beta NTI and other algorithms like this to determine which processes are driving the communities in time or in space, or in our case, a chronosequence, which is substituting space for time. In this way, you can directly tie environmental factors like soil pH or redox gradients, subsurface geology, et cetera, or biological processes with each community assembly process. So we care because we want to know who are the most important species for a potential specific function. So that would be looking at the key species that are occupying these niche spaces and seeing what's their capacity for biological interactions of interest that would inform our consortia engineering, potential consortia engineering for agriculture, oil degradation, or gut microbiome applications. Another thing that you can do with this community assembly analysis is to monitor the communities to see if they're stable or actively changing, and then tie that to which processes are driving them. So this one application would be to look at your changes in ecosystem function and potentially restore degraded habitats, which is what we were trying to do with our study, to see if that was a potential application. And as we all know, there's many complex communities consortia, and I just wanted to throw out the possibility of using community assembly analysis as another tool in your toolbox, not just networks, which could help to understand these complex communities like biocrust, biofilms, or the rhizosphere. So with that, I want to say thank you for your attention and make you think about what assembly processes could potentially inform your communities and how would that be relevant to your research. Thank you.
Okay, so thank you very much, Juliet, for the talk. It's really nice. I just want to hear what your idea would be for applying this kind of community assembly analysis process to a microbiome engineering experiment, for example, where you are intentionally driving the assembly of targeted microbiome. How do you think this would fit into this kind of research?
Intentionally targeting. So your question is, how would it tie into intentionally targeting some kind of community assembly, pushing the community one way or the other?
Yes.
Right. So I think the most interesting thing about community assembly analysis is that you can subset your data, right? So I didn't do this in my paper, but other papers have done it on soil pH, on rare taxa, on a bunch of different metrics that you can subset, and you can see what is the majority that's contributing to a stochastic or a deterministic process. So that would be most interesting to me, to see what the causes are of the change in your community, because there are so many variables that can contribute to that.
Yeah, thanks, Juliet, for this nice talk. And I think you added a very important point to the overall discussion here. This is the community assembly stuff. And I would like to link it to the keystone stuff with some sort of combination of question or comment. So assume you have a community which changes over time, which is actually a process of community assembly over time. Assume a taxon which is present throughout the entire time, but only to specific time points it functions as a keystone. So the question to you, to the audience, would be, how can community assembly help trying to understand under which conditions a present taxon becomes a keystone, so birth of a keystone or death of a keystone, but not birth or death of the taxon itself. I think this is something which sooner or later should be discussed in all this keystone issue stuff.
Thank you, Johannes. Yeah, I definitely agree with you. I think all of these analyses, the network analysis, the keystone taxa analysis, the community assembly analysis, they have to be backed up with some kind of functional study in the end, right? So I think, yeah, it's important to look at what's your unit. So instead of looking at one specific microbe whose behaviors can change over time, what's your microbiome doing as a whole in your specific environment? And yeah, I really liked your input. So yeah, thank you.
So he's junior group leader at Department of Environmental Microbiology in Helmholtz Center for Environmental Research, UFZ in Leipzig in Germany. And today he will talk about relationships between keystone species, ecologic theories, and predictive analysis.
Hi there, can everybody hear me? Is that a yes or a no?
Yes, I hear you and I see your screen.
Thank you. So first, I would like to indicate that this workshop has been really well prepared. It's a lot of different perspectives and important points regarding keystone species. And therefore, I will not do any introduction on the topic because they have been this much better done than I could do. So as when I was talking to Pamela about what would be the 5-minute talk that I could give on this topic, is what would be the relationship between keystone species and core microbiome with ecologic theory, and how we can use this to actually do predictions. Okay. The first point that we need to see there is that microbiomes and keystone species, they behave in different scales. And everything that I have been doing in the past 7 years with regards to predictive analytics is based on conversations I had and the material written by Dr. Sabine Hossenfelder. And she was the person writing the book Lost in Math, and she discussed exactly the disconnections between predictions and experimental data in the field of astrophysics and particle physics. And everything that she does is divided in levels of resolution. Okay, those scales that we know microbiome has. And the first one would be the high resolution. And when we're thinking about high resolution for microbial communities, this would be how a single species or even a strain actually behave in that community. And then we would have the medium resolution level that will be where we find the keystone taxa and the core microbiome. And evolving from these, we have the low resolutions that actually will be the ecosystem functioning and services. Remembering with a small reminder that ecosystem services are something that we believe are important in the ecosystem and not necessarily what is important in that ecosystem. And the message that I would like to give to you today is how these things connect with predictive analytics. And just to present my team, okay, I don't have it, so everything that I do is thanks to the people I work with. So this is the team I lead. Some people moved away. I have more PhD students, some PhD student graduates, so I'm not going to talk about this now. I have to actually update the picture. But I would like to talk about what's the basis for predictions and control of ecosystem functions. Starting with the predictions. So core microbiome and keystone taxas, they actually, with an interaction going from keystone taxas to core microbiomes to the different ecosystem functions, actually relates with the mechanism controlling functional capacity. And all of these is one field within microbial ecology. And then and this is the part where I really think that this workshop is amazing because it puts a lot of different people talking about the same topic. So national and international networks are very important to do these things. And also we saw here a lot of talks that indicate that, there is exploration of different systems. Over and over, we saw the interactions between experimentalists and modelers, and it's very important. And we also saw some type of flexibility that goes from one system to another. I will not talk about machine learning and the definitions because this is something that you can check in several different publications. I will be talking about the publication here in the middle a little bit, and then I will give a little bit of food for thought because it's just 5 minutes. So in this case, and this is something that all of you are going to be very used. The topic necessarily doesn't, so I'm talking about the publication in the middle there. We started with this type of things where we see differences between 2 data sets. In this case, we had 2 bioreactors, and they're different bioreactors, but they behave the same. And under different conditions, they also behave the same. But what would be the things that we can actually differentiate here to find the key taxas there? And we decided to use machine learning for that. And with the definition using machine learning, we managed to, using the taxa defined by machine learning, we managed to calculate the productivity of different types of carbon compounds produced in those bioreactors. And a very interesting thing, and we can talk about this later, is how we actually do these two independently, the two bioreactors, and the data still looks good. So a very important thing, think about experimental design and your ecosystem process and your data. And although things can look very bright, we did not manage to predict everything that we wanted for the system. And if I have to indicate that using machine learning with microbiology data is usually something that works 50% of the time, not all the time. And one extra thing is that and I think this was the interesting part, that our bioindicators were able to actually, all of them, because we also did metagenomes in the same places where we had the amplicon sequencing, and we recovered those genomes. And every bioindicator had mechanisms that actually participated in the process that we are looking at. And so success is not a guarantee. And I would like to give you, just as we have 5 minutes, a couple of minutes
1 minute, yeah, sorry.
that we have to think about. Big data in microbiology is unstructured and unbalanced. So that means we don't have structured data. Our data looks a lot like this. And unbalanced means that many times the things that we are interested, they should look more like this. So they should be more balanced, 50-50%. And most of the times we are using things that 90% of the data is actually not the part of the question that we look for, and we should try to change that. Experimental design is everything. So if you don't think about experimental design before you start the experiment, you are going to fail. It's very important to connect hypotheses to our ecosystem services. So ecosystem services is not something that the systems have. Those are things that we believe they have and that will be important for them. But remember, its evolution may have worked in a different way. And we should try to, if you want to do predictions using big data and using microbiology data, we have to think of the degrees of freedom. So if we imagine a soil microbial community, you are going to have thousands of ASVs or units of data features. If you're having a metagenome, this might go to the hundreds of thousands. And if you want to do big data, the amount of data samples you have should be 10%, at least more than 10% of the features you have. So that means if you have 5,000 ASVs, you should have at least 500 samples in that data set. And that means if you want to analyze the functional potential in that data set, using that you should have at least 500 metagenomes. And this is something that usually we don't think much when we are planning this type of experiments. And this was my presentation today. I'm sorry if I talked a little fast, but I wanted to give a short message, a couple of short messages. Thank you.